Hello, everyone. Welcome to the new Trent Seaman Center for Health Education at Duke University School of Medicine. I'm really happy to share this short tour of some of my favorite places in the human brain with you all. And this is a bit of an insider's look at the human brain. I'm going to share some of my biases with you about what intrigues me about the brain. And since it's something of an inside story, I thought, I should probably go to some inside place to film this. So we are in the specimen prep room for the Duke University School of Medicine for the laboratory courses that we teach with human brain specimens. And as you can see behind me, we have quite a number of human brain specimens in our collection. This is the place where we store, where we curate the specimens, and where we prepare them for our teaching. So in front of some of my favorite specimens, I'm just pulling one out and give you a chance to see up close and personal, as it were, some of the parts of the brain that especially intrigue me. So here's a brain from our collection, and I hope you can see it well. So we're looking at the anterior aspect of the brain, the right hemisphere, the posterior aspect, the left hemisphere, the dorsal surface, and I'm sure you can see the mid-sagittal plane there, where we have our longitudinal fissure separating the right hemisphere from the left hemisphere. And then on the ventral surface, I think a really nice demonstration of the ventral surface of the human brain, including the brain stem, the blood supply that enters the brain from its inferior surface, and the cranial nerves. Well, some of my favorite places in the brain. I would have to begin with the central sulcus. The central sulcus in the right hemisphere is this sulcus that we can see working its way from the dorsal midline in the superior longitudinal fissure all the way out to the lateral fissure. And I want you to notice the shape of this central sulcus, because I'm going to show you the left hemisphere, and you'll see that even in the same brain, the corresponding regions that you might think would reflect the symmetries of the body really can be quite different. So there's the right central sulcus, and now here's the left central sulcus. One feature that I think is quite different is that in the right side, there's a very distinct omega shape in the middle of the central sulcus, whereas in the left hemisphere, the central sulcus is much more straight. So here's the left side, and here's the right side. And my fingertip is right at that omega shape that I referred to. Well, that omega shape is roughly where the hand is represented in the somatic motor and somatic sensory cortex, with the somatic motor cortex being on the frontal lobe side and the somatic sensory cortex being on the postcentral side. I spent quite a few years working with a wonderful mentor, scientist and friend, Dr. Dale Purvis, and together we explored asymmetries of this region of the human brain, comparing the right hemisphere with the left hemisphere. And we were particularly interested in the notion of human-handedness and whether there was an asymmetry of the brain that related to human-handedness. Well, our studies were followed up by others who pursued this question, and it does indeed seem as though in the cerebral cortex there's more circuitry devoted to the preferred hand than the non-preferred hand, which raises some interesting questions, such as, is brain like muscle? Is bigger better? Does more skill necessarily mean more circuitry? Or does better skill simply mean better circuitry, not necessarily more circuitry? Well, these are fascinating questions that have been with us as long as people have been interested in studying the human brain. And that journey led me to explore this region of the human brain once upon a time ago. Well, more recently, my interest has been in the back of the brain. Here we have the occipital lobe, and the occipital lobe is home to our primary visual cortex and a series of associational visual areas that elaborate more and more abstract features of vision. I can't show you the primary visual cortex in this specimen because we'd have to look on the mid-sagittal plane, but I can show you roughly how visual information is processed in the occipital lobe. Signals first reach the medial portion of the cortex that forms the calcarine sulcus. That's our primary visual cortex. And from there, information generally is passed through a series of interconnected cortical areas, some of which tend to stream down the inferior aspect of the temporal lobe. We call that the temporal processing stream. And these areas on the ventral temporal lobe are especially concerned with recognizing the objects, the places, and even the faces that we encounter in our daily lives. For example, your face, or the face of your family, or the face of a loved one, is encoded in the areas that we find on the inferior surface of the temporal lobe. And unfortunately, there are people who have had strokes or other kinds of brain injuries affecting this part of the brain. And as a consequence, some of these folks lack the ability to recognize familiar faces. It's not that they're blind, it's that they fail to understand the significance of the visual stimuli that comprise a human face. Well, visual information is also sent along a more dorsal trajectory into regions of the parietal lobe and regions around the junction of the posterior temporal lobe, the occipital lobe, and the parietal lobe just above. This region of the occipito-temporal parietal junction is where we find some interesting areas that are especially concerned with motion processing. In fact, there's a region probably right about here in the human brain that corresponds with an area well studied in primate animal models called area MT, for middle temporal visual area. And this area is famous in neuroscience circles for being enriched in cells that have very sharp and precise preferences for direction of motion. This seems to be a region in the visual parts of the brain where our understanding of motion, or as our chair in neurobiology here at Duke likes to say, where we build our notion of motion. That would be our chair, Dr. Steven Lisberger, who gives wonderful talks on that subject. Well, the visual cortex is a part of the brain that I studied in my career after leaving the central region and exploring questions related to human-handedness. I moved on to animal models and studied aspects of vision and the organization of circuits in the visual cortex that pertain to understanding the fundamental aspects of visual perception, such as the orientation of the shapes and the lines and the angles that we see in our visual world, and the patterns of motion that either we generate as we move through the environment or objects in the environment that are in motion generate as they pass across our retinas. Well, that's a bit of personal history. Let me move on and just highlight some areas of the brain that are of really great interest to neuroscientists today and over the years, and some areas that are especially hot in today's world of neuroscience. Well, we're looking at the left hemisphere, so I should highlight some of the language areas that seem to be especially lateralized in the human brain. One that you probably know about is called Broca's area. Broca's area is found right about here, which is in the posterior portion of the inferior frontal gyrus. So Broca's area is a region that helps us produce speech, whether it be vocal speech or written speech. And although this is not well studied, probably written speech using our digital media that we use so often now to express ourselves in words. There are other areas that are specialized for different aspects of language, such as understanding language, be it in spoken speech or in written form. And those areas are distributed in the lateral superior temporal lobe, although most likely there is a broad network of brain structures that are distributed around the lateral temporal lobe and perhaps even into the inferior parietal lobule that are important for understanding speech. Classically, we call this Wernicke's area, but now I think we understand that it's not a single area corresponding to a cytoarchitectonic region, for example, but it's probably a network with multiple nodes that are encoding various dimensions of language for the purpose of understanding language. I know many of you listening to me are bilingual or multilingual, and for you, you very well may have different nodes that are important for understanding your various languages. And for those of you who utilize American Sign Language, yes, you probably have a node somewhere in here that encodes your visual understanding of language as expressed in the motions of the hands and the face. Well, let's turn over to the right hemisphere. Indeed, we've got corresponding structures in the right hemisphere in the position of the posterior part of the inferior frontal gyrus and in the superior part of the lateral temporal lobe. We conventionally don't call these areas Broca's areas and Wernicke's areas because if there were injuries here or if these areas were dysfunctional, there would still be speech and understanding of speech. However, something would be lost. Probably what would be lost is the ability to imbue speech with emotional tone and the ability to understand tone. Well, here in the United States of America, it happens to be Valentine's Day. And while we're thinking about making speech in the right hemisphere, I can't help but to think of the stories in the literature of marriages that have been broken over damage to the right hemisphere, specifically in the region of Broca's area. Imagine this: one spouse declares to another, "You don't love me anymore." Of course I love you. Why don't you believe me that I love you? Well, the idea here, and I'm not replicating this very well, but one spouse no longer believes the words by themselves because the other, who perhaps has had a brain injury to the right inferior frontal gyrus, can no longer imbue speech with passion, with the emotion that is the life force of our human relations. So although right hemispheric lesions are ineloquent, as sometimes we hear the neurosurgeons say, they can be nevertheless devastating to the human condition. Well, lastly, I want to show you a fascinating part of the brain that we'll come to near the end of medical neuroscience. And it's this part of the frontal lobe that sits directly above the orbits of the eyes. This is called the orbital cortex, and it's a part of the brain that was damaged in a famous case in the medical literature from the 19th century. I'm sure many of you know about this case. It's the case of Phineas Gage, who almost unbelievably survived a horrific injury where about a 13-pound, nearly meter-long iron rod was blasted through his skull, producing significant damage to the orbital and medial part of the frontal lobe in a region that we call the prefrontal cortex. Well, much has been written, much has been said over the years about the change in Phineas Gage's personality, his demeanor, his social mores. The fact is, we just don't know much of what is truthful about Mr. Gage following his injury. But there is a fascinating literature that's growing, examining people that are with us now who've had injuries to this part of the brain. And what we're learning about them is that their decision-making faculty has been impaired, especially decisions that pertain to their personal self and decisions that affect what they themselves are invested in emotionally and socially. And so this is a part of the brain that's critical for the integration of emotions and cognition in such a manner that it allows us to predict the future. That is, to weigh the consequences of our decisions and to act accordingly. So a fascinating part of the human brain that you'll be hearing some from me about later in the course, and continues to attract great interest from neuroscientists around the world who are trying to understand the networks and the circuitry and the physiology of this part of the human brain. Well, lastly, I want to show you the brainstem. This is really a wonderfully complex part of the human brain. Many neuroscientists are interested in this part of the brain. Sadly, many more are not. But for those of you interested in pursuing a career in healthcare, the brainstem is a critical part of the human brain, not only for its functions, but for what understanding the brainstem can tell you about your patients while you're proceeding with your interviews, your histories, and your physical examination. We have a number of beautiful cranial nerves that are visible here. I doubt you can see very many. In this perspective, it's really not my intention to show you those nerves here. We have an entire tutorial devoted to the cranial nerves, so I'd refer you to that. Nevertheless, we're going to try a bit of manual brain zoom to see if you can track the cranial nerves and see it as closely as possible. Okay, well, I'm nearly done showing you some of my favorite places in the human brain. Let me just conclude by quickly going over the 4 lobes of the cerebral hemispheres, which I hope will soon be very familiar to you as you view our videos of actual brains, as well as refer to our tutorials with illustrations and pictures. And for those of you who've purchased the textbook, you can look in the appendix and read about the anatomical organization of the cerebral hemispheres there. Okay, so looking at the right hemisphere, we have the frontal lobe to the anterior side, the occipital lobe to the posterior side, and in between the frontal and the occipital lobe is the parietal lobe in the superior aspect of the hemisphere and the temporal lobe in the inferior aspect of the hemisphere. The dividing line between the frontal lobe and the parietal lobe is the central sulcus. My index finger overlies the precentral gyrus, my middle finger the postcentral gyrus, and the space between my fingers representing the central sulcus. So the central sulcus is the division between the frontal lobe and the parietal lobe. The division between the parietal lobe on the occipital lobe depends upon what we can see from the mid-sagittal plane. Right about at the position of my finger is the parieto-occipital sulcus, which marks one point of demarcation between the parietal lobe and the occipital lobe. The other is a feature of the inferior aspect of the hemisphere called the preoccipital notch, which is attributable to a flap of dura that sits between the cerebellum and the occipital lobe called the tentorium. So an imaginary line between the parieto-occipital sulcus and that preoccipital notch is what divides the parietal lobe from the occipital lobe, and then the temporal lobe in front of the occipital lobe. So our temporal lobe then extends forward. Well, thanks for spending a few minutes behind the scenes with me in our specimen prep room in our Trent Seaman Center for Health Education. I hope you enjoyed this. I know I did. And believe it or not, I've got to teach a class in just a few minutes, so I'll sign off for now. Thanks for being with me, and I'll see you next time. Hello everyone. Welcome to Medical Neuroscience, and welcome to our first tutorial on the functional microanatomy of neurons. As we think together, I have 3 learning objectives for us to consider. First, I want you to be able to describe in very basic terms the classes of cells found in the central nervous system. I want you to be able to focus on one particular class that will spend much of our time in Unit 1 of this course considering the neurons. And I want you to think about the functional microanatomy of neurons. Now, I'll spend this tutorial explaining what I mean by that, but essentially I want you to be able to differentiate between the different parts of a typical neuron in terms of their structure and their function. And then lastly, I want you to be able to describe how that microanatomical structure of a typical neuron is compounded many thousands of times into the structure of neural tissue. And when we think of neural tissue, we're basically talking about gray matter and white matter. Okay, well, let's begin by looking deep into the brain. And to do that, I'm going to show you a slide that I prepared some years ago through the motor cortex of the human brain. So we're looking at just a few hundred square microns of tissue that has been stained with a particular dye called thionine to reveal the presence of what we call Nissl substance, which is basically the rough endoplasmic reticulum, or the machinery that's making proteins within cells. So it's just a great way to look at the cells that are present in tissue and to appreciate something about the composition of this tissue. So we're looking here, and we can see a variety of different types of cells. And indeed, the brain is a very complicated place, even from a histological perspective. So there are a variety of types of cells here, and I think we can roughly categorize them into three types. There are neurons, which are the primary processors of neural signals. And as I mentioned, we're going to spend a lot of time together thinking about neurons. There are neuroglial cells, which would be the subject of the next tutorial. And neuroglial cells, or just glia for short, perform a rich variety of functions. And just to summarize those functions, I would say that they support the electrical and the chemical functions of neurons. And then, of course, the brain being a highly vascular structure requiring a constant supply of blood, and in that blood comes nutrients such as glucose, oxygen, and the ability to eliminate waste substances like carbon dioxide. So with that rich supply of blood that we find throughout the central nervous system, we'll find vascular endothelial cells in brain tissue as well. So let's focus now on a neuron, and I'll make a few general remarks first, and then we'll get on with our survey of the microanatomy of a typical neuron. So neurons are the fundamental unit of function in the central nervous system. This is a very important point, and this is the reason why we'll spend so much of our time and energy in Unit 1 thinking about how neurons actually function. Now, for those of you who have a bit more background in the field of neuroscience or are more in tune with contemporary thinking in the field of neuroscience, you'll realize that some might challenge this dogma that's been with us since the debate of Ramon y Cajal and Camilo Golgi more than a century ago. So with the more modern discussions for the aficionados acknowledged, I would continue to assert that the fundamental unit of function in the central nervous system is indeed the neuron. Now, neurons possess all the metabolic machinery that are common to other somatic cells. So there's a region of the neuron that contains the nucleus and many of the organelles that are necessary for cellular life. So in that respect, neurons are very much like other somatic cells. But in several important respects, neurons are quite distinct. Neurons come in a rich diversity of form, and we call that form morphology. So when one sees diversity of morphology, you should be thinking that there might also be diversity of function or physiology, and indeed that's the case, as form and function and structure are intimately connected within the central nervous system, even at the level of individual brain cells. Neurons have unique bioelectrical properties that distinguish them from most, but not all, other somatic cells. For example, the machinery that's necessary to generate electrical signals in muscle cells is quite similar to what we see in most nerve cells. And finally, there are specializations for intercellular communication that are pretty unique to the nervous system. And for many neurons, most neurons in the mature central nervous system, this involves the secretion of special chemical molecules called neurotransmitters. Now, again, neurons have not evolved this capacity independently of other kinds of cells in the body. There are other secretory cells that are well known in other kinds of tissues, but the challenge of intercellular communication demands some pretty interesting and some pretty unique specializations that we'll come to shortly. All right, now let's survey the different parts that make up a typical neuron, such as the one illustrated here, and this will give us a chance to think about what I consider to be the functional microanatomy of neurons. And let's begin with the cell body. So neurons have a cell body, which, as I mentioned, contains all the essential organelles, importantly including the nucleus, that are common to other somatic cells. And for most neurons, this cell body is actually quite rich in organelles because of the incredible synthetic capacities that are required for neurons to maintain their morphology as well as to support their function. So we find an abundance of endoplasmic reticulum, both smooth and rough, for example, for the synthesis of lipid molecules and proteins, respectively. We find an abundance of mitochondria for generating the enormous energy supply that's necessary to keep neurons functioning within their homeostatic states. So, for a variety of reasons, the cell body is a very important component of the neuron. Now, from that cell body grow out numerous different sorts of protoplasmic extensions. And I'd like to draw your attention to one set of extensions, which are typically short, about 100 microns in length, and they emanate in all different directions from the cell body. These are called dendrites. Now, dendrites are very important for the microanatomy of a neuron because what they do is that they extend the surface area that allows the cell to receive inputs from other neurons. So wherever we see dendrites, we think of this as an input zone because dendrites are the means by which cells receive synaptic contacts from other neurons. Now, some synaptic contacts are made directly onto cell bodies, but for the most part we find them on dendrites. Now, dendrites also have some very peculiar, very fine microstructural features. And these are called spines. So if I can illustrate just the length of a dendrite for you, I'll show you what I mean. Imagine that's a dendrite, and if we were to look at very high power, we might see under a microscope that there are these tiny mushroom-shaped protrusions out the side of that dendrite. These are called spines. Sometimes they're not quite so mushroom-like in their shape. They're more of a filament that extends some short distance away. And sometimes there seems to be a little stalk and a protrusion at the tip of that stalk. So collectively, these yet increase the surface area of even a single dendrite and provide a place for synaptic contact from other neurons. So wherever we see a spine, we can be quite confident that there is a synapse in one state of formation or another from the axons that are surrounding that dendrite. Now, not all dendrites grow out spines. Some dendrites are smooth. And where we see cells that grow out smooth dendrites, we know that these cells tend to be those that have an inhibitory effect on the cells that they make synaptic connections to. So the presence or absence of spines on dendrites is one means by which we can classify neurons. And we know that that classification based on the morphology of the dendrite gives us some insight into the functional properties of those cells. The morphology of dendrites is one way that we can differentiate various classes of neurons, even among those that do in fact grow out spines. One of the very interesting types of cells that we find in the cerebral cortex is called the pyramidal neuron. And it's called that because the cell body is shaped something like a little pyramid. And from that cell body grow out a series of dendrites. And from the apex of the pyramid is one rather thick, stout dendrite that we call the apical dendrite, because it grows out from the apex towards the surface of the brain. At the base of the pyramid are a variety of other dendrites called basal dendrites. And this special apical dendrite might have some unique properties with respect to its role in receiving signals from other neurons, and those might be somewhat distinguished from those in the basal dendritic tree. So the distribution of dendrites is, again, another important way that we have of classifying different kinds of neurons. Some neurons are multipolar, and these neurons give rise to dendrites that seem to just emanate in all different directions without necessarily growing out a single apical dendrite. So again, another example of how dendritic morphology can give us some insight into the structure and physiology of different cells in the brain. All right, so let's continue our survey. Now, growing out from the cell body is a very particular protoplasmic extension that has unique properties that allow it to generate electrical signals. And this process is called the axon. Now, what's illustrated here is a fairly simple picture of what a typical axon might look like. It's a single process often that grows out from the cell body and can grow for some distance. Some axons are short, being less than 100 microns, and others are very long. In fact, some are incredibly long. Consider, for example, a cell that might have its cell body in the motor cortex right about here inside your head, and that cell has to grow an axon all the way down through the brain, through the brain stem, and into the spinal cord. Perhaps some length in the spinal cord, maybe even to the lumbar or sacral segments of the spinal cord. So that could be maybe a half a meter or more, depending upon your height. Or consider the motor neuron in the spinal cord itself that's going to send an axon out in a spinal nerve to innervate a muscle. If you're a very tall person, that axon might be close to a meter in length. Really an amazing dimension, considering the fact that the cell body itself is probably close to about 50 microns in diameter. So that gives you a sense of how much biosynthetic capacity must be built in to these neurons, as well as some insight as to their need for the energy and the raw materials that's necessary to grow out and maintain the function of that axon. Now, on the function of that axon, this axon has the ability to generate an electrical signal. It's a special signal that we call an action potential, and we'll talk more about that in a later tutorial. That action potential is regenerated along the length of the axon, and in that way it propagates from cell body towards the terminal ending of that axon. So for that reason, we think of the tissue that contains the axons as a conducting zone and allows for the flow of information from cell body towards axon terminal. Now, at the end of these axons, that's where we find our synaptic terminals, or synapses. And these are specialized contacts that allow one neuron to transfer a signal to another. They come basically in two varieties. We'll say more about these in a later tutorial. There are electrical synapses that have specialized junctions that allow charged molecules to pass directly from one neuron to another, and with it is conveyed the electrical signal. Other kinds of synapses are chemical, which is that there is a space or a gap or a cleft from one neuron to the next, and that cleft is breached by a chemical that's synthesized and released by one neuron, diffuses across that space, and then interacts with receptors on another. So there's a chemical message that mediates the electrical signal from one neuron to another. We'll have a lot to say about synapses, how they work, what are the important regulatory mechanisms that control synaptic activity as we progress through Unit 1. So much more on synapses as we go. Now what I'd like for us to do is to consider how this brief survey of the microanatomy of a single neuron can be compounded many tens of thousands of times in the structure of neural tissue. Now imagine tens of thousands of neurons arranged in parallel, very much like the one we're looking at here. And what I want you to appreciate about this parallel organization is that where we find the cell bodies and the dendrites, that is the input zone of one neuron, we'll find the input zone of many thousands of neurons that are arrayed in parallel in tissue, such as the cerebral cortex. And we have a name for this. We call this gray matter. So as we begin to look into the inside of the human brain, I'll be highlighting for you various divisions of gray matter. Those are distinguished from white matter. And now we can think about the cellular components that help us define what exactly do we mean by gray matter and white matter. So gray matter is where we find the cell bodies, the dendrites, and the synaptic connections of neurons. White matter is the conducting zone. That's where we find axons and the special insulation forming cells that surround those axons called glial cells, specifically in the brain the oligodendrocyte. So that is another component of white matter. And then of course both gray matter and white matter require nutrients in the form of vascular supply, so there are vascular endothelial cells in both kinds of tissue, both gray matter and white matter. And then finally, let me again emphasize that where we find the output zone of one neuron, we find the input zone of many others. So gray matter is both input zone and output zone, depending upon which neuron we have in mind. Okay, well, enough illustrations. Let's look at some actual brain cells. So here are some pyramidal neurons in the cerebral cortex that I prepared myself. And what we're looking at is a stain that shows us beautifully a pyramidal-shaped neuron here. There's its cell body, sort of the pyramid looking something like that, and a thick, stout apical dendrite progressing off towards the surface of the cortex, which is out here somewhere. Here's another pyramidal neuron and its thick apical dendrite. We also see some basal dendrites extending out away from the cell bodies here. Now, we also have an axon, at least one that I can recognize, and it is this process right here. It's extending away some distance away from that cell body. So that's the means by which this particular cell is going to generate electrical signals and then propagate those signals to its neighbors, some quite near, and then some at a great distance away. Here's another pyramidal cell in the visual cortex, but we don't see its apical dendrite because we're looking down from the top, so its apical dendrite is actually right about there. And then here's its cell body, and from its cell body are all these beautiful branching basal dendrites. Now, if we look very closely at these dendrites, and I know this image is not quite as clear as we would want it to be, we can see some spines on these dendrites in that region and here again down there. So if we have a careful look at one such area, I think you'll be able to appreciate that there are in fact some small little mushroom shapes, some filaments that are extending away from those dendrites. And so this would be an example of a typical pyramidal neuron that grows numerous spines along its basal dendrites as well as on its apical dendrite, but typically a little bit further away from the cell body. Okay, here's another view at a large collection of pyramidal neurons, and we can see again their cell bodies. There are little pyramidal shapes here, apical dendrites heading towards the surface of the brain, and then lots and lots and lots of multipolar basal dendrites going off in all different directions. Now we also can detect the presence of axons that are coming out and extending down into this white matter. That's what all these thin filaments are that we can recognize heading down through this relatively unstained background tissue. So here we see all the various components of the functional anatomy of a neuron, the dendrites, the cell bodies, the axons. Well, we can't quite see everything because we haven't yet seen the synaptic terminals. And to give you a sense of that, I want to show you yet a different view of brain tissue. So this is tissue that's stained with a green dye that has labeled axons as they make their way through brain tissue, specifically the gray matter of the cerebral cortex. And I would just draw your attention to these little points of light, these little spots that we can see along many of these axons. So these little points of light are actually places of synaptic contact. Now, some axons, they would grow out a terminal ending, and then there'd be a synaptic junction at that terminal ending. Others tend to make this beaded type of structure, and that's what we see here along these green axons. These are called synaptic boutons, and they're making what we say en passant, or in passage, synaptic connections. So along these boutons there may be synaptic contacts with dendrites out along the sides here. I know I drew them as if they look like they perhaps are spines, but they're really boutons because this is an axon, not a dendrite. So that's what we see here in these little points of light, synaptic boutons. Here's one last view of neurons and their axons. So we see cell bodies of retinal ganglion cells and axons that are growing out away from them towards the optic nerve head. And these are axons that are going to form the optic nerves that allow the retina to connect up with the brain and send visual signals into visual processing centers in the brain. Now let's return to some illustrations of different kinds of neurons and recognize some of the various classes of cells that are present in the nervous system. We've talked a fair amount about the corticopyramidal cell already, and this is a great example of what we call a projection neuron. And the reason why we call it that is because, as I've alluded to, it grows out an axon that can then project for quite some distance within the central nervous system, maybe hundreds of microns, maybe even millimeters, tens of millimeters, maybe even tens of centimeters, depending upon whether the cell is projecting within the brain or whether it's growing out an axon down through the brainstem and into the spinal cord. So projection neurons are called that because they grow axons that project outward, away from the cell body, over a considerable distance. And these cells often are excitatory, meaning that they have an excitatory effect on their synaptic partners. They make their synaptic partners more likely to generate their own electrical signals. Now, in addition to the long axon that projection neurons send often at great distance away, I'll just point out, as is illustrated here, that the axon can actually give rise to more local branches that can activate cells that are sitting right next to the cell of origin of that axon. So there are local connections as well as a single axon that projects over a great distance. Now let's look at these corticopyramidal cells in context. Here to the left we have our illustration of the individual pyramidal cell, and now we can see the pyramidal cell that is present within the cortex itself, cell body, apical dendrite, basal dendrite, and then a long axon. And here are two other corticopyramidal cells. So this is a typical view of what a projection neuron looks like and where it might be found in the brain. So the pyramidal cell is a projection neuron. It sends axons over considerable distances that have the effect of exciting their synaptic partners. They may also give rise to local collaterals that might just excite their near neighbors as well. So they sort of have a dual function of activating local cells, but also cells at a distance. Now, in addition to these projection cells, we can also recognize some neurons that send axons over a much shorter distance, only perhaps 100 microns or a few hundred microns. One example is found here in this cortical stellate cell. So the cell has a cell body. It's typically multipolar. There are dendrites that go off in different directions. And then there's an axon. And that axon may branch over the nearby territory within the dendritic tree of that cell or just beyond it. So this is called an interneuron because it doesn't project very far at all. So within the same bit of gray matter, we can have projection neurons and we can have interneurons. Now, the interneurons come basically in two physiological classes. There are those that excite their neighbors, and then there are those that inhibit their neighbors. And they're roughly equally divided, with about as many excitatory interneurons as there are inhibitory interneurons. If we look into the spinal cord, we can also see examples of projection neurons as well as interneurons. Here's an illustration of a very simple circuit that we have throughout our spinal cord and in the sensory and motor elements of our brainstem as well. And this is an illustration of the knee-jerk reflex, also known as the myotactic reflex. So we have two kinds of projection neurons that are involved here. We have a sensory neuron with its cell body in the dorsal root ganglia, associated with our spinal nerves, very close to the spinal cord itself. And it has a long axon that runs out through a spinal nerve, and in this case, innervating a sensory structure embedded within the skeletal muscle itself. This dorsal root ganglion cell grows a central process of its axon that enters the spinal cord. Now, it's worth pointing out this particular type of neuron to make the following point. This sensory axon is sending signals into the spinal cord. For that reason, we call it an afferent neuron, sending information centrally. There's another type of projection neuron here in the spinal cord, and it's called the motor neuron. It has a multipolar cell body in the spinal cord, and it grows an axon out to the muscle. And the motor axon is sending signals away from the central nervous system to the muscle. And for that reason, we call it an efferent axon. So get comfortable with this difference between afferent and efferent. Afferent means signals coming in to the central nervous system, and efferent meaning signals going out of the central nervous system. Sometimes we use these terms to talk about the flow of information with respect to the cell body itself. We can think of the dendrites as being afferent because information is coming in towards the cell body, and we can think of the axon as being efferent because signals are going away. Now, we also have within the spinal cord a short axon interneuron, and that's found right here. So this interneuron has the effect of inhibitoring the antagonistic motor neuron, so that when our hammer tap hits our patella tendon, the quadriceps can contract while the antagonistic muscle, the hamstring muscle, is going to be more relaxed because of the suppressive or inhibitory effect of this interneuron on the motor neuron that supplies that hamstring muscle. Okay, well, this concludes our survey of the microanatomy of neurons, as well as a brief discussion of different classes of neurons. I hope it's helped you understand the different parts of neurons, what they look like, how they function. So to help you consolidate your understanding of all of this, I'll leave you with just a couple of study questions. Concerning what we've been talking about as the functional microanatomy of a neuron, I want you to think about how does information flow through a neuron? And think about the various parts that are listed out in these response options, and go ahead and select your choice. And then lastly, let's take this concept and compound it and apply it to understanding the structure of neural tissue, specifically in this case white matter. I want you to think about what are the cellular components that we find in gray matter and white matter, and in this question consider those components found in white matter and choose your appropriate response option. Okay, well that concludes our first tutorial. I'm sorry I went on about as long as I did. I will try to be more concise in the future, but I hope you enjoy thinking with me about these topics. I know I enjoy sharing them with you. So on to our next tutorial, considering the non-neural cells that make up the central nervous system. See you next time. Welcome to this tutorial about the non-neuronal cells of the central nervous system. We have 3 learning objectives today. We want to, again, simply describe the basic classes of cells found in the central nervous system. And now, in this tutorial, we want to focus specifically on the glial cells. So I want you to be able to describe the basic functions of the 3 types of glial cells found in the central nervous system. One of those types of cells has an important role to play in forming a particular kind of physiological barrier called the blood-brain barrier. So we'll spend a little bit of time thinking together about that, and I want you to be able to discuss it or characterize it with a friend or a study partner. So let's begin by again looking deeply into the brain as we did in our first tutorial. Here again is a view of the motor cortex as seen in a histological slide that reminds us that, again, the cortex is a very complicated place. There's lots of different kinds of cells that are found here, and specifically what we find are neurons, which was the subject of our first tutorial, neuroglial cells, and that's what we're going to focus on today, or sometimes we'll just call them glia for short. So we'll talk in more detail in just a few minutes about all of their various functions. And I would just remind you again that there are also vascular endothelial cells found in this tissue as well, given the fact that the brain requires such a steady and abundant supply of the cardiac output in order to maintain the functions of the brain. Okay, so let's think now about the neuroglial cells. And the neuroglial cells provide a variety of functions for brain tissue. Perhaps the broadest summary statement that captures a variety of their functions would be to simply state that they support the metabolic and the signaling functions of neurons, which often involves both electrical and chemical signals. Neuroglia participate in the formation of neural circuits and in a variety of forms of synaptic plasticity. This is a very exciting new aspect of glial cell biology that's just now beginning to open up, and we're coming to see glial cells as really being active participants in the process of making new synaptic connections in the brain. Certain types of glia, they make myelin, which is a form of insulation around axons that aids significantly in the propagation of electrical signals along this component of the neuron. Glia contribute to the formation of the blood-brain barrier. We'll have more to say about that at the end of the tutorial. Glia participate in the inflammatory response in injured neural tissue, and that includes the phagocytosis of cellular debris. Other types of glial cells contribute to the formation of scar tissue in damaged brain and spinal cord, and so this is part of the way that the brain responds to injury. All right, now let's consider different types of glial cells, beginning with what is in some respects the most complex of the 3 major types of glial cells, the astrocyte. So here's an illustration of an astrocyte from your textbook. And astrocytes are primarily found in gray matter. And the reason is that they're closely associated with the cell bodies, the dendrites, and the synaptic connections of neurons. And as you'll recall from our first tutorial, these are all cellular components that are found in gray matter. Glial cells help to maintain the ionic balance of the extracellular fluids that surround neurons, and that's very important for maintaining the capacity of those neurons to generate and maintain electrical signaling. One additional role of astrocytes is to take up and process neurotransmitters. Those are the special chemical molecules that are released by presynaptic terminals in the process of intercellular communication from one neuron to the next. So astrocytes have a role to play in the processing of neurotransmitters. As I mentioned, astrocytes are the glial element that assists in the formation of new synapses and new circuits within gray matter. This is, again, a newly discovered property of astrocytes, and it's one that is opening up new opportunities for research, as well as potentially new therapies for intervention for individuals who've had damage to their brain or spinal cord. Astrocytes are that category of glial cell that contributes to the formation of the blood-brain barrier, as well as the barrier between the brain and the ventricular system within the brain, called the brain ependymal barrier. And astrocytes are that type of glia that can differentiate into a type of cell that can form fibrillary scar tissue within injured brain. And so this is important if there has been injury, and that injury is evolves in time to form, let's say, a small cavity. As cellular debris is removed, then that cavity can be filled up by astrocytes that will grow in and form a kind of scar tissue to occupy that space in damaged brain. Okay, the next type of glial cell to consider is the oligodendrocyte. And the oligodendrocyte is more of a specialist. Its functions seem to be much more limited than that of the astrocyte. Specifically, the oligodendrocyte is found in white matter because this is the cell that forms the insulation around axons in the central nervous system. And that insulation is called myelin. Now, if we were talking about a peripheral nerve, in the peripheral nervous system, we'd be talking about a different type of myelin-forming cell called a Schwann cell. So I just wanted to make that distinction between the oligodendrocyte and the Schwann cell. They both make myelin, they both insulate axons, but the oligodendrocyte is the one found in the brain and the spinal cord, whereas the Schwann cell is found in the nerves of our body. So what good is myelin? Well, myelin aids in the propagation of neural signals along myelinated axons, and we'll say more about that in just a moment. But before we do, I'll also mention that the oligodendrocytes present antigens that influence the outgrowth of axons both in the developing brain and in the recovering brain that's undergoing a process by which it will try to regenerate connections that have been lost due to injury. Now, unfortunately, the oligodendrocyte also presents antigens that are subject to immunological attack in certain diseases of the central nervous system, the preeminent example of which would be multiple sclerosis, in which oligodendrocytes are the subject of the immunological attack, and myelin can break down. And with the breakdown of myelin is a loss in the efficiency of the conduction of neural signals along axons, and that can lead to neurological dysfunction. Okay, let's come back to this point about oligodendrocytes making myelin and what that does for the efficiency of information transfer along the length of an axon. So here's an illustration from your book showing you two portions of an oligodendrocyte that are coming together, forming myelin. Myelin would be this wrapping of membrane that surrounds this axon here. So these many concentric rings of membrane, that's what we call myelin wraps or myelin sheaths. And so here is the cell body of one oligodendrocyte. There'll be the cell body of another somewhere off screen here with its nucleus. So these two oligodendrocytes are coming together, and there's a small gap between them called the node of Ranvier. Okay, so what these oligodendrocytes are doing is that they're insulating axons by generating layers of membrane that wrap around those axon segments. And this has the important function of decreasing the leakiness of the membrane of that axon. And so this allows the passive flow of current along the length of the axon to be much more efficient. Now, as I mentioned, there are gaps between these myelin segments contributed by different oligodendrocyte, and those gaps are called nodes of Ranvier. And these nodes are quite important because they provide a place whereby ion channels and ion pumps can be concentrated. And so the benefit here is really twofold. The electrical signal is regenerated here in this node of Ranvier, and the signal can be regenerated across these segments of myelin that are contributed by different oligodendrocytes, and the signal essentially can jump from one node to the next. We call that saltatory conduction. More on that in a later tutorial. But there's another advantage of having nodes of Ranvier, and that is that one can populate these nodes with the proteins in the membranes here of the axon with great efficiency. So you end up having to create and populate fewer ion channels and pumps in a smaller region of axonal membrane than you would otherwise if there were no myelin. One would have to have channels and pumps along the entire length of that axonal membrane in order to keep that electrical signal propagating. But with nodes of Ranvier and myelin segments between the nodes, we can be much more efficient, both in the electrical properties of the conducting tissue, but also in the biosynthetic requirements to create that machinery that's necessary to generate those signals. So these are all great advantages of having myelin insulating axons. Okay, and our third type of glial cell that we'll talk about in this tutorial is called the microglial cell. Now this is a really fascinating cell. It's actually a cell that is a special type of mononuclear phagocyte that resides in the central nervous system, although it's actually derived from hematopoietic precursor cells that migrate into the brain during early embryonic development. So these cells enter the brain and they take up residence in one of two forms. In the dormant state, this microglial cell is called a ramified cell, and that's what's illustrated over here to the left. So this is a ramified microglial cell. It has lots of filamentous processes that extend away from the cell body. And here below we have a photomicrograph that shows a population of ramified microglia. Well, these ramified cells are dormant. They are lying in wait, and in the event of injury or inflammation, then these cells will retract their processes and convert to what's called an amoeboid state, which is what we see over here in the right-hand figure. So these are all activated microglial cells that are now on the move. So the amoeboid form is the activated mobile state when microglial cells are mobilized and engaged in phagocytic activity. And this is one principal function of microglia, is that they phagocytize cellular debris that is found at the site of injury in the nervous system. But they also secrete signaling molecules, numerous kinds of cytokines, for example, that can modulate local inflammatory responses. Now, one further type of glial cell that is worthy of some mention is actually not a mature cell at all. It's a stem cell. So within the central nervous system, we now know that there are stem cells that have the capacity to multiply and to differentiate. And these stem cells resemble glial cells of the astrocyte and the oligodendrocyte forms. So Here's an illustration of glial stem cells. And these seem to be a subset of immature astrocytes that are located near the ventricles, often adjacent to blood vessels, as we see here in this illustration. And these glial stem cells can give rise to more stem cells, or they can mature into astrocytes, oligodendrocytes, and even neurons. So these cells exhibit the key properties of somatic stem cells. They can proliferate, they can renew their populations, that is, they can form other stem cells, and they have the potential, the potency, to make all the cells of a given tissue. In this case, of course, we're thinking about the central nervous system. Now, there's also a glial stem cell that more resembles the oligodendrocyte, and this oligodendrocyte precursor is found scattered throughout the white matter of the central nervous system. And when this cell differentiates, it mainly gives rise to mature oligodendrocytes, although under certain conditions it might give rise to astrocytes and perhaps even neurons. Okay, now let's return to this concept of the blood-brain barrier as we conclude today's tutorial. The blood-brain barrier is a specialized permeability barrier between the capillary endothelium and the extracellular space within neural tissue. And it's formed by tight junctions between capillary endothelial cells. So, for example, here is the nucleus of a capillary endothelial cell, which is otherwise wrapping around, forming a capillary wall. And where the processes of this cell overlap, we find the histological feature known as tight junction. So tight junctions seal up the spaces between these overlapping regions of this endothelial cell as it forms the capillary. Surrounding this endothelial cell are the foot processes, or the end feet of astrocytes, that completely envelop this capillary endothelial cell. So together, the endothelial with its tight junctions and the astrocytic foot processes form a limiting barrier around the vascular compartment. So this has some very important consequences. This barrier excludes large water-soluble molecules from freely diffusing into the central nervous system, as well as certain pathological microbes and certain toxic compounds that might otherwise enter the brain. So a very important protective function formed by this limiting barrier. Of course, there are some molecules that must enter the brain, such as glucose and a variety of amino acids. Such molecules typically are transported across this barrier by both active and passive means. Now, unfortunately, this barrier also provides a limitation for certain pharmaceutical agents that we might want to provide patients in the healthcare setting. So, certain drugs, for example, might be engineered in a way to circumvent this barrier, because otherwise passage of such compounds would not be facilitated by the presence of this blood-brain barrier. Now, across the lifespan, this blood-brain barrier is present in most places throughout the central nervous system, with a few important exceptions. Essentially, wherever there are special populations of neurons that are secreting proteins that need to enter the blood in order to have a hormone effect throughout the body, then this blood-brain barrier tends to be porous. That is to say, the tight junctions are not present, and there are openings that allow substances to diffuse across this junction between brain and bloodstream. And so that's important, for example, at the base of the forebrain, where we find the hypothalamus, where there are neurons that are secreting peptides into the blood that will affect the anterior pituitary. There are other places in the brain, such as in the pineal gland, where hormones such as melatonin are secreted and enter the bloodstream. So wherever there is a hormone secretory function, there is going to be some loosening of this blood-brain barrier. But with those exceptions, this barrier is present at birth and it's maintained throughout the lifespan in normal, healthy brain. There are some diseases and some conditions that can affect the integrity of this barrier. And when that integrity is challenged, then the brain is more susceptible to the entrance of molecules that might alter its physiology and decrease its function. Well, let's conclude this tutorial with a study question. I want you to think about a patient that has had a stroke, and that stroke causes some small region of the brain to be damaged. And I want you to think about the glial cells that are present in the brain around that region of damaged brain, and think about which activity of glial cell do you think happens first. Well, think about these activities and the sequence in which they might unfold, and go ahead and record your response. Well, thanks for your attention. I hope you enjoyed this tutorial on the non-neuronal cells of the central nervous system. For our next tutorial, we'll get back to talking about neurons and focusing on the means by which they generate electrical signals. I'll see you then. In order to understand the human brain, it's helpful to know some of the terms that we use to describe both location and direction when it comes to brain anatomy. So let's just go over some of the very basic terminology. Well, here we have an adult human brain, and the front of the brain is here to my left, and the back of the brain is here to my right. So the front part of the brain we call the anterior end, and the back part we call the posterior end. So anterior is towards the front of the brain, posterior is towards the back. Now, of course, the front of the brain is also towards the nose, so for that reason we also call this direction rostral. So anterior and rostral mean the same direction for the adult human brain. Now, towards the back end of the brain, we call that caudal, although for reasons that we'll mention in just a moment, that word caudal, meaning tail, doesn't exactly fit the forebrain, but it does fit when we include the spinal cord. So we'll get to that in just a moment. So anterior rostral towards the front of the brain, and posterior caudal towards the back of the brain. Now, if I turn the specimen so that you can appreciate the mid-sagittal plane, dividing the left hemisphere to the right hemisphere, I think it's easy to appreciate how we talk about medial to lateral in brain space. So medial is towards the midline, lateral is out towards the side. And that's true for all levels of the central nervous system. Now, sometimes which way is up can be just a bit confusing in the brain. When we view the brain from the lateral surface, the top part we can refer to as the superior surface of the brain or the dorsal surface of the brain. The lower part we can refer to as the inferior surface or the ventral surface of the brain. But these terms get just a little bit confusing when we look at the mid-sagittal plane in a different hemisphere. So let's do that. Now, with the mid-sagittal hemisphere in hand, we can talk about position as it applies not just to the forebrain, but also to the brain stem. Well, as I mentioned, towards the front is what we call anterior or rostral. Towards the back of the brain is what we call posterior or caudal. But notice what happens in the relationship between the hindbrain and the forebrain. There is a flexure or a bend that takes place in the axis of the brain as the forebrain develops on top of the middle brain or mesencephalon. So all of this forebrain seems to be running in the plane of the table, whereas the brain stem is off at an angle, about a 120-degree angle relative to the plane of the forebrain. That's what we call the cephalic flexure. And below that point of flexure, these positional terms are applied in a different way. So when we talk about caudal for the forebrain, we're talking about the posterior part of the occipital lobe. But now if we talk about caudal relative to the brainstem or the spinal cord, we are talking about the inferior direction towards the tail, the cauda equina segment of the spinal cord, which is the most inferior stretch of the spinal cord. So towards the tail is quite literally towards the horse's tail or the cauda equina when it comes to the brainstem and spinal cord. Now, what about dorsal and ventral? So for the brain stem, dorsal means towards the back. And if you can imagine this brainstem extending into the spinal cord, towards the back would be on this aspect of the brainstem. So the cerebellum is dorsal to the brainstem. Posterior for the brainstem is synonymous with dorsal. So if we talk about the posterior aspect of the brainstem, we're talking about its dorsal surface. Likewise, if we're speaking of the ventral surface of the brainstem, we're talking about the part of the brainstem and spinal cord that's towards the belly, or is ventral. Ventral is synonymous with anterior when it comes to the brainstem and to the spinal cord. So this can get a bit confusing. Again, just to review a few of these terms, when we're talking about anterior relative to the forebrain, that would be in this direction, towards the front of the brain. Anterior with respect to the brainstem would be right in this location here. Ventral for the forebrain would refer to the inferior surface of the hemisphere. But ventral for the brainstem is synonymous with anterior. Now finally, superior and inferior continue to mean towards the top of the body being superior and inferior is towards the lower part of the body. So we can say that the pons is superior to the medulla and the pons is inferior to the midbrain. So superior and inferior are similar, as is medial and lateral. If we were to look along this mid-sagittal plane, the cut surface is medial, whereas the side surface of the brainstem is what we would call lateral. So medial and lateral are the same for the brainstem and spinal cord as they are for the forebrain. So with some knowledge and some practice with these positional terms, it will make great sense to you and will have the capacity to discuss relative position within the brain as well as among these superficial features with some precision that can help us align the central nervous system with the rest of the body.